Mrs. President, Mrs. the Rector of the University of Iceland, Mr. the Dean, Mrs. the Ambassador of France, Madame l'Ambassadrice. It's a real privilege and honor for me to be present here for this 100th anniversary of the University of Iceland. Thank you very much for the invitation. Thanks also for all who organized my visit here. I'm particularly pleased to participate to this symposium on the legacy of Björn Sigurðsson. You just heard about his contribution, and when I was a young student myself in retrovirology, I remember, of course, learning about his discovery of this slow infection that turned out to be caused by lentiviruses. So it's a real pleasure to be for the first time in Iceland, where he has made this work. Indeed, I am coming myself from I started my study at the university in the late 60s, and I was not thinking at all at that time that I will work on viruses or on infectious disease. Why? Because I show you on this slide that in the early 60s, indeed, it's this statement from Björn saying that one can think of the middle of the 20th century as the end of one of the most important social revolutions in the history, the virtual elimination of the infectious disease as a significant factor of social life. So in turn, when I started my study at the university, my interest, curiosity, motivation was certainly for other major and little human pathology, and at that time in the 60s, we were talking a lot about cancer and leukemia already. So when I got the opportunity to start at the Keldur Institute for preparing my PhD, I had the chance to work in a laboratory interested in cancer and leukemia, but they were using retroviruses as a model to study cancer and leukemia. And this is the history of retroviruses, very much summarized, let's say, on this slide, indicating that indeed retroviruses are known since the early 90s, as you can see here. But it has been a very strong interest, particularly in the United States in the early 70s. Indeed, when I started at Pasteur, it was a special program funded by United States on relationship between retrovirus and cancer at that time. You can see on this slide that already in the 70s, the hypothesis of a provirus was stated by Temin in 1969. It was the first identification that retroviruses can carry a gene called oncogene, and at that time we were speaking about viral oncogene. In 1970, our team in Baltimore reported for the first time the reverse transcriptase activity. That was certainly a turn, let's say, in science, changing the dogma that indeed your DNA can be transformed in RNA and then in protein. But for the first time, they demonstrate that RNA can be transformed in DNA by an enzyme, the reverse transcriptase of retroviruses. And we had to wait the end of the 70s with the discovery that indeed what we thought it was a viral oncogene in the retroviruses was indeed not a viral gene, but a cellular gene. At that time, it has been quite a lot of change in the field of cancer and leukemia, and I must say that most of the retrovirologists at that time moved from studies on retroviruses and turned to work mostly on oncogene. However, some of us, like me at the Pasteur Institute, continue to work on retrovirus and cancer and leukemia. And certainly the reason why we have been successful in the identification of HIV in 1983, as it has been mentioned. But certainly also we have to acknowledge the work of Björn Sigurðsson by describing for the first time slow infections that were causing in sheep encephalitis and pneumonia. And that turned out to the identification of the first lentivirus, Visna medi. And you heard that HIV and those viruses in sheep are indeed from the same family. So when you look at the history of retroviruses, some of them have been related to cancer and leukemia and lymphoma. Some of them have been related to immune defects, inflammation. You heard just about the contribution of Sigurðsson mentioning the role of this slow infection causing inflammation. Inflammation due to persistent infection. I will come back on that point because this is true for HIV as well. We know that this virus can cause autoimmune malignancies as well. So this is a family of viruses that can cause not only cancer or leukemia but also other human diseases that are among the diseases that cause a lot of problems to our society still today. Let's go back for a few minutes, 30 years ago, almost the right anniversary dates, June 5th, 1981. That was the first report by Michael Gottlieb and colleagues in the United States of the first case of pneumocystis carinii associated with a profound immune depression in 5 young homosexual patients in the United States. If I'm showing you this slide, is certainly to mention that there's the first sign of an emerging epidemic, and that reminds us that Burnett sentence was indeed not true, and since then we learned that indeed infectious diseases are still around and they will be around forever probably. But if I like to mention and to show you this slide, it's to tell you that for us basic scientists, it's very important to be in contact and in relationship with others, epidemiologists, clinicians. They were the first to report these new symptoms. And it's really through their observation that we started to guess that this disease, this new emerging disease, was probably due to an infectious agent transmitted by blood and blood derivative because of the first case of AIDS detected, as you can see on this slide, in hemophilic patients. The first observation also indicated that this disease was probably transmitted by sexual route because of the infection identified first in homosexual, but also later on few cases of heterosexual transmission indicating that the virus can be also transmitted from the mother to the child. So this clinical observation and epidemiological observation were certainly critical, and ourselves at the Pasteur Institute, it's really by the clinician that we have been mobilized ourselves. They came at Pasteur Institute at the end of 1982 for asking a very, very simple question. Did you hear about this new emerging disease? And we say no. So they explained to us. And they said, do you think a retrovirus can be causing this new emerging disease? We asked, why do you think so? And they said, because there is this new human retrovirus reported in the United States called HTLV, reported by Bob Gallo and colleagues, and this virus is infecting T lymphocyte. We say, that's true, but HTLV1 is a transforming virus. And you are just telling us that the patients are losing their CD4 lymphocytes, since one of the major observations is a profound depletion of CD4 cells. However, we say, look, there is some other retroviruses, like feline leukemia virus, that induce immune deficiency in cats, and very often cats are dying from immune deficiency before dying from leukemia. So we told them, we can check. Let's check whether it's a retrovirus or not. We don't believe about HTLV, but it can be another retrovirus. At that time, we were not thinking about slow infection, I must say. And we started to ask a very simple question as well: when shall we look for a retrovirus? Where? And how to look for this virus? So it's really by discussion during a meeting that we decide to take a very classical strategy. Since we did not think HTLV could be the virus, we said, okay, we are going to try to isolate the virus from a culture, from a sample from a patient. But we told our colleagues, we don't like to try to first try from a blood sample, because you just mentioned that the patient do not have any CD4 cell. So can we have a patient at an earlier stage? And they say, yes, at earlier stage they have what we call lymphadenopathy, unexplained generalized lymphadenopathy. And of course we immediately react and saying, that's it. If there is a virus, the virus should be in the lymph node. So indeed, this was the start of another very close collaboration. Basic scientists, clinician, epidemiologist, but also the patient themselves. They ask a patient whether he will accept to participate to the research. And one patient kindly accepted to have a biopsy, a lymph node biopsy. We put in culture the cell from the lymph node, and we learned later on that was the right decision. You can see on this slide that we learned 10 years after, in 1992, on the right of this slide, how many cells can carry HIV in the lymph node, the black point by in situ hybridization. So certainly it was the right decision to have a sample from the lymph node, and we decided also to look into the culture, a culture that was made possible because of the recent identification at that time of a factor that was called T-cell growth factor, now known as interleukin-2. So we were able to grow the cells in the presence of interleukin-2, and 2 weeks later to start to detect in the supernatant of the cell culture a reverse transcriptase activity. A reverse transcriptase activity that was functioning in the same condition of HTLV1. But this virus did not have the characteristic of HTLV1. So that was the report in Science on May 2019, SP3, where we reported, as was mentioned for the first time, the isolation of the virus, a virus that we call at that time lymphadenopathy-associated virus, since it was identified from a patient with generalized lymphadenopathy. It was not from a patient with AIDS, but with a patient at risk for AIDS. We showed in that paper that we were able to propagate the virus on peripheral mononuclear cells and on cord blood lymphocytes as well from donors. I just mentioned the condition of the reverse transcriptase activity, but when we started to compare the protein of the virus that we just isolate with reagent obtained from Bob Gallo for HTLV1, we did not found any cross-reactivity between the major protein of our virus, P25, and the major protein of HTLV1, both by different techniques. It was no cross-reactivity also with another HTLV1 protein, and we showed also in that paper the presence of antibody against the virus just isolated in a second patient. That was with AIDS. In that paper, we were showing also a picture by electron microscopy. We did not detail anything about the morphology in that publication because we were ourselves surprised. The particle was looking like a retroviral particle, the size, the envelope, but the core of this particle was not classical core of oncoviruses. So we started to discuss with others, we started to look back in the literature until we found out that this morphology was exactly the same as the morphology of Visna midi. So it's how we started to think that this virus LAV was a member of the lentivirus family. However, this first work was certainly not sufficient to convince the scientific community and the authorities as well that this virus LAV was the etiological agent of the AIDS. We had to make some more work to link the virus itself and the AIDS disease by making more viral isolate from patients with AIDS, by making serological investigation, epidemiological investigation, by making the characterization of the virus itself as well as the characterization of the viral isolate. That means for us at that time, working on mammalian retroviruses and on other research topics, that we had to stop immediately any other research program in our lab. We knew also that we were only very few of us working on this topic. We said, okay, we had to mobilize others. We have to mobilize other clinicians, other researchers. We are virologists. We are not immunologists. We need to have molecular biologists to work with us and so on. So we started to mobilize others. It was not also easy to convince others to come and work with us. And meanwhile, and in parallel, we knew that we had to develop very urgently serological tests for diagnosis, because we started to be convinced that this virus was the cause of AIDS, but we knew that this virus was causing AIDS in hemophiliac patients that was receiving blood derivative. So we had to mobilize also the private sector as well, and we established a very strong and efficient partnership with the company, Sanofi Diagnostic Pasteur, in order to develop those tests. So this research, since the very beginning, I said, started by clinical observation, epidemiological observation, go to the laboratories. It's really a research from the bedside to the bench and return to the bedside since the very beginning. Today we are speaking a lot about translational research, and I'm sure that Sigurðsson himself was already making translational research on his time. Indeed, we are doing translational research for HIV since the beginning, with the identification of the virus and the development of the first diagnostic test in 1985, available for the prevention of blood transmission. We also started to try to make counseling in order for people at risk to be tested for this virus in order to avoid mother-to-child transmission and sexual transmission, but at that time we did not have anything to propose in terms of treatment itself for those people. We started to characterize the virus itself, the tropism of the virus, and show that the CD4 lymphocytes were the first target for the virus. And we were very naive at that time, because we observed into the culture that the CD4 lymphocytes, when they were infected by this virus, they were dying. We thought that's it. We understand everything now. The virus infects CD4 cells and they die. So it's how we can explain the profound CD4 depletion. I'm saying it's very naive because still today we are far to understand all the mechanisms of the CD4 depletion in HIV infection. We identify the receptor, the first receptor for HIV, the CD4 molecule, and we learn later on that indeed there are also co-receptors. We started to characterize the replication cycle of the virus and the reverse transcriptase of this virus, and that was certainly very useful for the development of the first antiretroviral treatment. The first antiretroviral drug was AZT that was used as monotherapy in the mid-80s. However, we learned very quickly that AZT alone was not working with the emergence of resistance to AZT. However, AZT was shown to be already very efficient to prevent mother-to-child transmission. And we had to wait 1996 to have the combined highly active antiretroviral therapy known under the name of HART, which brought so much benefit for the patient. Today we know, after many years after the beginning of treatment, that patients are still on life. We have been able to reduce the mortality by more than 85% in the patients because of the development of this combined therapy. Meanwhile, we continue also to characterize the genome of HIV, LAV at that time and to compare to other viruses. And we started at that time in the early 80s to identify the genetic diversity of the virus. But this first data on sequencing and identification of the diversity of the virus was very useful for developing new monitoring tests for measuring the viral load by PCR, quantitative PCR, real-time PCR, and also to study and to monitor drug resistance in the patients. The cloning and sequencing of the virus was done between 1984 and 1985. We published the sequence in January 1985. We understood at that time that it was a unique and very complex genome, distinct from HTLV1, but comparing to Visna and Visna medivirus, we found out, and equine infectious anemia virus, we found out a very similar organization. So that was the final proof, let's say, that HIV was a member of the lentivirus family. As I said, we also understood that when you compare one virus isolate to another isolate from another patient, the virus are not exactly the same. We started to understand the diversity, and a lot of work has been done since then to study the diversity and the origin of HIV worldwide. You know that HIV, the first virus isolated in 1983, is known to be a member of the HIV-1 type, and we know that this family of HIV-1 is subdivided in different groups, the group M, N, O, and more recently the group P of HIV-1. Very soon after the identification of HIV-1, a second type of HIV was isolated and turned out to be found to be named later on HIV-2. We learned by comparing the different viruses to a monkey and to primate virus in Africa that both HIV-1 and HIV-2 originate from introduction into human of primate lentivirus. You see on this slide that we first report ourselves the relationship, the genetic relationship between HIV-1 group M and SIV from chimpanzee in Cameroon. We know today that HIV-1 derived from chimpanzee viruses, and the virus from the gorilla is probably deriving also from chimpanzee infection. We learned also that HIV-2 itself is deriving from introduction into human in West Africa of a virus from the sooty mangabees, with a very close genetic relationship between both the monkey virus and the human virus. As you can see also in this slide, by phylogenetic studies, the authors have been capable to estimate the date of introduction of the different viruses into human, leading to the beginning of the epidemic of group M virus between 1915 and 1941, for the group O virus between 1990 and 1940. So we have now clearly information about the diversity, but also the origin of the virus. It has been a wonderful effort at international level to understand better the complex interplay between the virus and this host. And we know that we have to consider both the genetic diversity of the virus, but the genetic diversity of the host as well, for the response of HIV infection into the host. The tropism of the virus itself for CD4 cells, but soon after we learned that there is also target for HIV, macrophages. Macrophages, which are also the main target for Visna meli. We learned that indeed all antigen-presenting cells, including dendritic cells, are target for HIV. We know that the replicative capacity and the tropism of the virus can depend on its diversity. We know that the virus is capable to induce and to have immunosuppressive capacity. Certain protein of the virus, like TAT, for example, but also the glycoprotein of the virus, have immunosuppressive capacity. And again, it could be some variation according to the virus itself. We know that HIV is capable to induce abnormal activation signaling pathways, and this is certainly a critical issue to consider in HIV pathogenesis. On the host determinant, certainly we have learned a lot during the last decades or 2 decades regarding the factors that are involved in the life cycle of the virus itself, and this is very important for developing new therapeutic strategy. We learn also that the cells have intrinsic defense with restriction factors that has been reported. The first one are apobactin 5-alpha, tetherin, and or BST2. But the most recent one that was just reported last week in Nature, some HDA1. SARM-HDA1 is an interesting factor that is able to restrict HIV-1 infection in dendritic cells. It's not able to restrict HIV-2 infection or SIV-MAC infection. Why? Because VPX, one of the regulatory proteins that is present only in HIV-2 and SIV-MAC, is capable to bind to SARM-HDA1 and to induce the degradation of these cellular factors. These cellular factors is indeed a very important factor regulating type 1 interferon, which explains also why SARM-HDA1 can play a role in the innate immune defense against infection like HIV infection. So some of the factors that has been reported during the last decades are also mediators of host innate and adaptive immunity. And that has to be taken into consideration for the future of HIV research. All over the years, many targets have been used for developing antiretroviral therapy, and this slide is summarizing the different inhibitors that has been developed. The first one, of course, the reverse transcriptase inhibitors, followed by the protease inhibitors, and more recently fusion inhibitors, entry inhibitors, and integrase inhibitors. Today, we have more than 30 antiretroviral agents available in Western countries. More than 10 generics today are also available for resource-limited countries. And we have learned a lot about the power of this antiretroviral treatment. And this slide summarize the impact of the treatment not only on the mortality and morbidity in the patient, but also the impact on the prevalence of HIV infection, the modelization that has been done already a few years ago, showing that if you treat only 30% of the patients infected by HIV, you can see that on the top of the slide, it will be no effect on the prevalence of infection. Now, if you treat all the patients, you can see the blue line, we will be able to eliminate HIV infection in 250. So the reason why we are so much considering universal access to treatment for all. It's also for eradicating the epidemic as well. And as you see also on this slide, it's cost effective. Because if you treat only 30% of the population, you can see the cost, which is increasing all over the years. If you treat all, you will see over time a decrease in the cost as well. So this has to be considered for the mobilization, both for research and intervention everywhere in the world. There are decisions that need to be made by, of course, decision makers, politicians, for the benefits of all communities everywhere. But it turned out that from my own experience and the experience of others working with Africa or with other low-income countries, that if you want to convince the political leaders locally and the health authority locally, you have to provide them scientific evidences. And to provide scientific evidence, you have to have research as well, locally, because they will be convinced only by scientific evidence derived from their own country. So it's the reason why it's so important to try to strengthen those countries for making research and to link the research and intervention together. And so these two components, research and action, let's say, should be, of course, connected, associated with training, strengthening capacity, and strengthening of infrastructure. You can see on this slide that is also an important component that we have been considering since the very beginning of the AIDS epidemic in the Western country, and is really starting to be mobilized now in low or middle income countries. I'm mentioning the HIV community, the participation of the HIV communities itself, the representative of patients. Of course, they have been very active, as you know, in Western countries for making decisions, for pushing the politicians to take decisions, for also decreasing the price of antiretroviral treatment by the pharma. The role of activists has been critical. And of course, they have to organize themselves in the developing countries, but they are starting today. And for me, certainly HIV has been the first example where scientists, health professionals, and communities has been really working strongly together to try to convince the decision makers. The resource available for HIV/AIDS has been very important, as you know, and it's through this scientific evidence that we at international level have been able to convince the United Nations to make efforts. I just summarize all the effort on this slide, and would like to mention, of course, the Global Fund for the 3 diseases of poverty, HIV/AIDS, but also tuberculosis and malaria, that was launched, as you can see in this slide, in the early 2000s. But there is other international effort like the Gates effort of the Gates Foundation, the PEPFAR program, the Bush program, the Clinton program, and so on. This wonderful effort at international level should nevertheless continue. In 2010, it was a year of replenishment for the Global Fund, for example, and an estimated promise of 12 billion was said at that time. But the estimated fund needed is indeed 18 billion of US dollar. And I can say even today that the promise of 12 billion will not come. So we are very much concerned today about the future. We are concerned about the future because even if it has been a wonderful progress, as you can see on this slide, within the next, let's say, 7 years, you can see that we multiplied the number of patients in the developing world on heart. You can see that today it's about 36% of patients that need to be on treatment, that are on treatment in the developing world. Is not sufficient since for one patient starting the treatment, there is more than 2 new cases of infection every year. So that's really not sufficient, and we have to know that we still have 30 million of people living by HIV everywhere in the world. HIV/AIDS remains today in the second position on the list of the deaths caused by infectious disease. More than 95% of these infections are in resource-limited countries. And this is an important issue to keep in mind, about 60% of HIV-positive persons do not know their serological status and can infect others. In Europe, for example, according to the country, between 15 to 50% of people ignore that they are HIV positive and can transmit to others. It's still a very sensitive topic because of sex addictions. There's a lot of stigma and discrimination still today. So HIV/AIDS 30 years after it's still there, we have new challenges. We have new challenges because in, let's say, in Western country, we can say that we are not speaking anymore about AIDS. We are speaking about people living with HIV. We have a chronic infection today on heart. In the South, we have, of course, still AIDS mortality. And we are starting also to have few cases of chronic infection on heart. So the differences between the North, let's say, the poor countries and the rich countries all over the year is slowly disappearing. But you can see that there are, in our country, we are starting on long-term treatment to see few complications associated to the treatment, like cardiovascular disease, 8% of patients on heart, on long-term heart. 15% of patients developing cancer, lymphoma in particular, liver disease, neurological syndrome, aging disease, osteoporosis, Alzheimer's disease in patients on heart. And of course, we need to have new therapeutic strategy for tomorrow to have a short-term treatment instead of having this long-term treatment, which is difficult for universal access in long-term, in low-income countries. And it certainly will be easier to propose to the patients a short-term treatment that will eradicate the infection. Of course, eradication is not an easy task, and personally, I don't believe about eradication of HIV. However, it's clear that we need new therapeutic, new vaccine approach, and for that we need to learn a little bit more about HIV pathogenesis still today. We have learned that, of course, AIDS is due to HIV, but there is two critical issues. The fact that very early after the infection, and this slide summarizes this point, very early after infection, there is an establishment of a persistent infection with the establishment of viral reservoir in the different compartments of the body. Persistence, like indeed all the lentiviral infection. We know also today that HIV is capable to induce inflammation and chronic immune activation. We said today that HIV it's a disease of inflammation. We know and we learn that everything is decided very, very early, even if it is a slow infection, everything is decided within the early days after infection, during the acute phase of infection. What are the mechanisms that result in the establishment of persistence, and what are the mechanisms that result in inflammation and chronic immune activation? We are far to understand everything. We have certainly in our hand tools to try to understand better how to protect against HIV/AIDS. Those models are summarized in this slide. We have in human and also in Asian monkeys a few individuals that are capable to control naturally their infection. We call them elite controllers. The elite controllers have undetectable viral load, and the level of the viral reservoir in their different compartments is very low. So what are the mechanisms by which they control naturally their infection? We are learning over the years that, of course, the genetic of the host is important. HLA-B25, HLA-B57 has been shown to be critical genetic factors. However, we learn also that a small group of elite controllers have a very strong and efficient CD8 response capable to eliminate the infected cells. But we have a small group of elite controllers that do not have strong CD8 response. But the sensitivity of their macrophage and the sensitivity of their CD4 lymphocyte is low. We are far to understand all the mechanisms, but we suspect restriction factors involved in the limitation of HIV infection and the limitation of HIV reservoir in these elite controllers. So we have models to try to understand control of HIV infection, and we have, on the other hand, models to understand control of inflammation and chronic activation. This is the African non-human primate, like Suti Magabi, like the African wind monkeys. Those monkeys do not develop AIDS. The virus replicates. The virus is not controlled in the monkey. What is the difference between, let's say, human or macaques that develop AIDS? In those monkeys, there is an attenuated chronic immune activation, an attenuated inflammation. So we have two distinct models, one to try to understand the mechanism of control of the infection, one is to understand the control of inflammation and of chronic immune activation. Regarding the reservoir, we are learning more and more. We know that where and what are the viral reservoir of HIV-1. We know that this reservoir is predominantly in T central memory cells. We learned more recently that PD-1 is a marker for HIV reservoir and can even maybe a target for purging the reservoir. We learned that there is differences in memory subset in gut compared to the periphery. We have a good correlation between HIV replication and immune activation in the gut of the patients. And we know that most latently infected cells in the gut, in the blood, are non-gut homing resting memory CD4 T cells. In addition to the lymphoid tissue and to the gut, we know that the central nervous system is also a reservoir for the virus, with a compartmentalization of HIV that has been reported in CSF and which is related to HIV-associated dementia. There is a correlation as well that has been reported between the level of astrocyte infection with the severity of HIV-associated encephalitis. Why HIV is persisting during heart? There is still some controversy, let's say, in the scientific community, but there is mostly, let's say, two mechanisms. One is remaining low level of viral replication that is occurring even on heart. And the second mechanism, which are going together, is the survival of T cells that carry latent infected virus and that proliferate. So these two mechanisms are certainly to be considered for trying to develop new strategy for targeting HIV reservoir. The latency, we know it's due to a block in transcription, and of course there is several strategies that can be used to try to unlock the latency mechanism. There has been already several drugs developed, like the HDAC inhibitors, for example, microRNA, for example, as well. Several approaches using immunomodulating agents like IL-2, IL-7 to try to boost the proliferation of the cells, to activate and to boost the proliferation of reservoir cells. An enhancement of anti-HIV immunity in patients on heart and to stop heart and then enhance HIV immunities. The intensity of the treatment and to treat as early as possible and then try to stop the treatment. And there is new proof of concept regarding gene therapy using zinc finger nuclease as an approach for gene therapy. Those approaches, some have been successful, some are still ongoing, but certainly we are learning better today, and we really think that it should be possible to obtain, let's say, not the eradication, as I said before, I don't believe too much about eradication, but I believe in functional cure. That's been reducing the level of reservoir, and the patient can remain with its virus, but with a control of the virus in the body. We believe also that a vaccine for HIV/AIDS should be possible, at least a vaccine that will block the evolution, the progression of HIV infection to AIDS. Why we believe it? It's because we have learned all over the years that the decrease of the viral load, the decrease of viral reservoir is clearly associated with a clinical benefit. Regarding the eradication, we have a proof of concept with this famous Berlin patient. This patient that is HIV positive developed leukemia and has been transplanted with stem cells from a donor that were carrying a mutation in one of the major co-receptors of HIV, CCR5. This Berlin patient is now living without treatment, and the virus is undetectable in this patient. We still don't understand the mechanism. We still don't understand the mechanism because this patient, Berlin patient, was also carrying viruses that were not only CCR5, but he was carrying viruses that infect cells through CXCR4, the second major co-receptor of the virus. So how to explain this eradication in this patient? This is certainly something to work on, and I know that there might be a second Berlin-like patient in San Francisco, and researchers are trying to understand the mechanism. I just mentioned the controllers that tell us that we should be able to control HIV infection, HIV replication. I mentioned also the African primates that are able to control disease progression because they control inflammation and abnormal activation. We have learned that there are few individuals that are protected against HIV infection. We call them exposed, non-infected individuals. They have partners and they have sexual relationship, unprotected sexual relationship, and they don't get infected. There are some that are on experimental vaccines that confer protection into monkeys infected by SIVs. And there is certain vaccine candidates that has been shown at least to induce immune response in vaccinees without major side effects. And finally, I would like to say that the results of the Thai RV144 vaccine trial was very encouraging for vaccine research. Certainly was not this vaccine trial was not telling us that we have a vaccine today. But for the first time, we had a 30% efficacy with a vaccine candidate that certainly was not optimal. However, we have to understand why this small proportion of vaccinees got protected in this vaccine trial, because one of the major obstacles for vaccine research today is that we do not know what are the mechanisms of protection and what are the correlates of protection that vaccine should induce. So there is a need for further research, both in vaccine and for vaccine research. There is a global alliance, we call it the Global HIV Vaccine Enterprise, that has been developed several years ago. For HIV cure, up to now, it was nothing, let's say, as an alliance at international level. So at the International AIDS Society, we decided last year to try to stimulate and to promote multidisciplinary science on HIV cure at international level by connecting researchers involved in developing an international scientific strategy. They are currently working on it, and we hope to have a global scientific consortium toward HIV cure by 2012 that will be supported by the main international organizations. Some of them have responded very positively, like NIH, for example, or AMFAR, or also the National Agency for AIDS Research in France, as well as SIDAction. So we are working on one side with the scientists and the other side with the funders in order to stimulate this research on HIV cure at international level. So it's why we are more or less in a mood to be optimistic today. We are in a mood to be optimistic also because we know that antiretroviral treatment can be efficient to protect. And the first example was to protect mother-to-child transmission. We have elimination of mother-to-child transmission in Western countries. We learned also during the last year indeed, that if you introduce antiretroviral drug into microbicide, so a new form of microbicide, you can use this approach to reduce the risk of transmission. This Caprisa study showing a decreasing risk between 40, let's say, and 50% using this new microbicide formula. We learned also that treatment as prevention in MSM can reduce by around 40% the incidence of HIV infection in homosexual. We learned all over the year, but even more recently, a few weeks ago, that antiretroviral treatment can reduce by 96% the risk of infection of partner if the positive partner is treated early by antiretroviral treatment. We learned that we can decrease also the prevalence of HIV infection in population strongly affected by HIV. There are several studies published, and so we certainly need to have a sustained commitment on this area. The future strategy to reduce the incidence of HIV infection in the world will certainly be combining different approaches, certainly still working on behavior changes, working on new biomedical strategy, including also development of vaccine and development of new antiretroviral strategy that as part of prevention today. We need also to work and to fight for social justice and human rights if we want to decrease the stigmatization and discrimination of population, which is a major and the first obstacle for testing and early treatment today. The response against HIV/AIDS is starting also to see some results and some consequences on global health. Just mentioned on this slide very few examples showing that in different countries in Africa, we are starting to see a decline in infant mortality and an increase in life expectancy. And I'm not speaking about HIV individual, I'm speaking globally. I'm speaking globally. We can see on this slide that non-HIV infants, for example, in eastern Uganda, there is a reduction of around 80%. 60% around reduction of child mortality in South Africa, in some part of South Africa. And there is also a substantial impact on taking care of HIV on TB prevalence, mortality, and morbidity in developing countries. So we should not stop our effort, because I'm personally convinced that what we are doing for HIV, TB, or malaria, for example, is very important for other diseases. And I just finished by showing you this slide that I told you that now in HIV patient on heart, we are starting to see non-AIDS related mortality, development of cancer, aging disease, cardiovascular disease, and most of them are also due to inflammation, abnormal inflammation or autoimmune malignancy or immune defects. So what we are learning for HIV can be also very useful for this new emerging disease in our society. Whatever the kind of research that we know, what things that I think we learn in the field of HIV is that if we want really to have impact on health, we have to work in a network of experts in different areas, from the private and the public sectors. We have, since the beginning, I was saying, worked very closely from clinicians as basic scientists. But this kind of networking of health professionals, basic scientists in different areas of basic science, together with experts in public health, experts in social economical science, working also together with communities representative of patients has been critical for the development and the progress in HIV/AIDS, and I'm sure this is the way to go for research in other areas for tomorrow. Certainly we have new challenges, new concepts today, and a new generation of players, but they have to keep in mind that it's really when we work together that we can be effective. Thank you very much for your attention.